Hello everyone, I am Dr. Tulsidas Patil. Today I am going to discuss about enzyme linked immunosorbent assay, which is one of the topic mentioned in Pharmaceutical Biotechnology course of third year B Pharm. Disclaimer: this video presentation is made for education purpose only and not meant for any commercial interest. The information provided in this video by the presenter is taken from his own personal experiences and understanding and from the references which are quoted at the end of this video presentation. Learning outcomes. After watching this video, students will be able to understand the purpose and working principle of ELISA technique, what are the different types of ELISA like indirect, direct, sandwich and competitive ELISA, and at the end they will understand various applications of ELISA technique. So let's understand about ELISA. ELISA is enzyme linked immunosorbent assay. It is one of the important immunological method for detecting and measuring antigens and antibodies. If we compare ELISA with the radioimmunoassay, there is very minor difference in between these two techniques. When we consider ELISA, in this antibodies are conjugated with the enzymes, whereas in radioimmunoassay, the antibodies are conjugated with the radioactive isotope. ELISA technique is very popular because of its high sensitivity, safety, economy, and its simple instrument requirements. Now let's understand the working principle of ELISA technique. In ELISA, a polystyrene plate, most commonly 96 well plates are utilized, which are coated with antigen or antibodies on its surface. The second component of ELISA are primary antibodies, which are having unique structure and greatest specificity for binding to particular antigens by lock and key mechanism. Third component of ELISA are secondary antibodies, which are linked with enzyme. Those act as a biocatalyst. Secondary antibodies either bind to Fc portion of primary antibodies or to the different epitope on the antigen. It is depending upon the type of material which is coated on polystyrene plate. Now, when we introduce the substrate specific to enzyme, there will be the reaction in between the enzyme and substrate, which will generate the colored product. This colored product is measured in terms of absorbance by spectrophotometric plate readers, and this absorbance is directly proportional to the amount of antigen antibody complex and in turn the concentration of target molecule. Now let's understand the working principle of ELISA by studying different types of ELISA. First, we discuss indirect ELISA. In indirect ELISA, we detect and quantify the antibodies which are present in serum or any biological sample against the particular antigen which is considered as a determinant of particular infectious disease, or this can be a tumor markers which are identical for particular cancer. So in this technique, wells of the polystyrene plate are coated with antigens and the serum samples or other biological samples containing primary antibodies are added in wells, and these are allowed to react with the antigens. So there will be the formation of antigen antibody complex, and if there is any free primary antibody is remaining, it is washed away. Now this antigen antibody complex is detected by introducing enzyme linked secondary anti-isotype antibody, which will specifically bind to primary antibody. Now if any free secondary antibody is present in unbound form, it is washed off and wells are incubated with substrates which are specific for enzyme. For example, we add alkaline phosphatase or horseradish peroxidase. So there will be the reaction in between enzyme and substrate, which generates a colored product which is measured in terms of absorbance. This absorbance is directly proportional to the concentration of the target molecule. The second type of ELISA is direct ELISA. It is same as that of the indirect ELISA. The only difference is that the enzymes are directly linked to primary antibodies instead of secondary antibodies. Primary antibodies will bind to antigen which is coated to the well of polystyrene plate. Any unbound primary antibody is washed off and directly we add the substrate which will react with the enzyme and it will generate the color. The third type of ELISA technique is sandwich ELISA. Now in sandwich ELISA, antibodies are immobilized in the wells of polystyrene plate and samples which are containing antigens are allowed to react with the antibodies which are coated in plate. So after we do the washing step, we add enzyme linked secondary antibody specific to different epitope of the same antigen and it is added and allowed to bind to that antigen. Now we carry out the washing step so that unbound secondary antibodies are washed off and then we add the substrate which is specific to the enzyme. This substrate and enzyme will react and generate the colored product which is measured in terms of absorbance. The fourth type of ELISA is competitive ELISA. Now in competitive ELISA, antibodies are initially incubated in a sample solutions containing antigens of interest. So as per the concentration of antigen present in the sample, antibodies will attach with the antigens and some antibodies may remain free. Now this antigen antibody mixture is added to antigen coated microtiter well. Now if more the antigens present in initial original sample, lesser the free antibodies available to bind to antigen which is coated in a well. When we add enzyme linked secondary anti-isotypic antibodies, they will bind to primary antibodies as in case of indirect ELISA. However, here the scenario is different. Higher concentration of antigens in original sample, greater will be the binding of primary antibodies to antigens prior to incubation in microtiter wells. So lesser primary antibodies will bind to the antigens present in the wells. So less complex will be formed in between primary antibodies and secondary antibodies. This will in turn lesser will be the generation of colored product and lesser absorbance. Means higher the concentration of antigen in prior sample, lesser will be the absorbance. There are wide range of applications of ELISA. If we see few examples, it is used to detect and measure the presence of antibodies in the blood. These antibodies can be autoantibodies or antibodies which are developed against particular infectious disease like hepatitis A, B, C or HIV. Then ELISA is utilized to detect and estimate the levels of various tumor markers like prostate specific antigen, carcinoembryonic antigen. It is used to detect and estimate the hormones level, for example, human chorionic gonadotropin or other hormones like luteinizing hormone, follicular stimulating hormone, prolactin, testosterone. ELISA is also utilized in detection of drug abuse. The drug abuse for various drugs like cocaine, amphetamine. These are the references which are used for making this video presentation. Thank you for watching this video.